This video is on the lymphoid pathway of hematopoiesis. Hematopoiesis is the production of all the cellular elements in blood. This occurs in the bone marrow. Hematopoiesis starts with a stem cell, an HSC. When the stem cell divides, one daughter cell will go back to repopulate the stem cell pool, and one will go on to differentiate. This daughter cell can become either a myeloid precursor or a lymphoid precursor. The HSC becomes the multipotent progenitor cell, or MPP. This cell can then become a myeloid progenitor or the lymphoid progenitor. In the lymphoid path, it becomes the lymphoid-primed multipotent progenitor cell, the LMPP, before progressing to the early lymphoid progenitor cell, the ELP. Here it can become a T-cell progenitor or continue down the lymphoid path to become the common lymphoid precursor, or CLP. The CLP can move forward to a T-cell progenitor, a natural killer cell, an ILC, a dendritic cell, or continue to become a pre-pro B cell. This pathway starts in the bone marrow but then moves to other organs before the cells completely mature. The first pathway is the simplest one. The natural killer cell, or NK cell, develops directly from the lymphoid stem cell at the CLP stage. The NK cell is part of the innate immune system that protects against pathogens and cancer. Although primarily produced in the bone marrow, there is some newer evidence that they can develop and mature in secondary lymphoid tissues. The NK cell is part of the innate lymphoid cells, or ILCs. The ILCs are separated into three groups according to the cytokines they secrete. Before moving to the T and B cells, we need to discuss the clusters of differentiation, or CDs. The way to distinguish between the T and B cells are by their surface proteins, which is termed the CD. The CD proteins are given numbers such as CD4 or CD8. This is to avoid confusion from naming each protein. There are currently over 300 CD protein markers known. T cells can develop from ELPs or CLPs. These progenitors leave the bone marrow and travel to the thymus, which sits on top of the heart, to become fully mature. Therefore, they are called T cells, T for thymus. In the thymus, the cells are called thymocytes as they are not fully functional T cells yet. Maturation requires several steps where the CD arrangements on the cell membrane surface change. The final product is either a CD4+ or a CD8+ T cell. T cells can further distinguish themselves as T helper cells, T cytotoxic cells, or T regulatory cells. The cytotoxic T cells are known as killer T cells, not to be confused with natural killer cells. B cells take about 1 to 2 weeks to develop. They start in the bone marrow, moving through a number of steps to become an immature B cell. The B cell then leaves the bone marrow and travels to a secondary lymphoid organ to mature. B cells can produce 5 different types of antibodies, but only one type of antibody can be made by one B cell. Once B cells fully differentiate, they are known as naive B cells until they are activated by an antigen. From there, they can become plasma cells, which secrete antibodies into the circulation, or memory cells that wait until future antigen exposure to produce antibodies. The lymphoid pathway for the HSC is a complex and complicated process for the acquired immune system to be fully functional. This concludes the video on the lymphoid pathway of hematopoiesis.